So a DNA binding protein I thought was pretty interesting was CREB. So the CMP response element binding protein is a transcription factor which activates the CMP response element present in over 4,000 genes in the human genome. So CREB is phosphorylated by a wide range of kinases, one of them being protein kinase A, which is a result of high CMP concentrations. Proper activation of CREB requires the binding of cofactors, activators, including CREB binding protein and P300. So some examples of pathways that require the CREB protein are the RAS system, the GBCR receptor, and even calcium-gated ion channels. After the downstream kinases phosphorylate the serine 133 residue, CPP using a 6-pix domain is able to bind CREB, which starts the formation of a CREB homodimer. However, for CREB to recognize its DNA element, it uses the P300 histone-acetyltransferase to form active enhancer elements. So the two main domains we can look at in CREB. The first is the kinase inducible domain, which is where the serine 133 is phosphorylated. After that, once the homodimer starts to form, the leucine zipper is what really holds homodimer together. Here we can see a 3D structure of just the leucine zipper of CREB. Looking at look closer, we can see the hydrophobic valine on the inside and ionic interactions between arginines and glutamates on the outsides to form specific leucine zipper formation. Looking at the DNA binding region, you can see non-covalent interactions between our protein and the atoms on the major groove. So with the help of the TATA binding domain, this homodimer is able to be constitutively activated and have increased binding affinity to its DNA element. As you've seen in the active structure, this leucine zipper has a slight left-handed twist formed by two parallel alpha helices. So CREB is involved in a lot of processes like inflammation, DNA repair, and immune response, and even cell cycle progression. Thus, you can expect that regulation of CREB is associated with various cancers like leukemia, melanomas, gastric cancer, and even brain cancer. Because one of the kinases responsible for activating CREB is the calcium depolarization activated calcium dependent protein kinase, CREB is involved in a lot of neural processes as seen here. Specifically, the dysfunction is also associated with schizophrenia and Alzheimer's disease. In order to treat cases where CREB is misregulated, KG501 was developed as an inhibitor to the CREB-CBP interface. KG501 was developed through a small molecule library screen, which is then validated by NMR.